What I'm going to do today actually is give you a sort of like an overview of sort of the work I've done in the past, and then tell you a bit about some of the work we've done sort of more recently, and then I'm going to give you a potential glimpse into what I think may be the future. I've always liked this slide here, this image. I always refer to this as, actually one of my postdocs, Eva, actually pulled this out. The hands-on neuroscience. So I think this is like a lovely sort of figure to start with. I shall just put this up. It sort of says what I need to say. I've been particularly interested in sort of tackling problems by not being worried about the subject area, the approach. I've been very pleased in my sort of career to be able to sort of look at things in lots of different ways, whether or not that might be a sort of biological approach, a chemical approach, and a physics approach. And I'll sort of give you a sort of brief sort of potted history of where I came from and probably why I actually take the approach I do at the moment. Here's my potted history of interdisciplinary research. So this is me at school, so it's sort of rather sort of grim sort of figure. I'm smiling actually, I always smile. I always think it's important to be positive. My school years were along the lines of, I did a lot of sports. Very interdisciplinary. You name it, I played it. Even I was actually even captain of fencing. I think I only ever sort of did it once, but I was rubbish at it, and so I lost my captaincy. This is for my mother, illicit winemaking. She doesn't know this. I used to make those were my first experiments, really, sort of under my sort of bunk bed. And I did a lot of exams. I went to one of those schools that sort of John Major absolutely hates. Frightfully posh and everything. But as you know, Chris sort of said at the beginning, one of the things, I wasn't born with a silver spoon in my mouth. Both my grandfathers left school when they were 14 years old. And my grandmother, she actually spent time in the poorhouse. But actually, as you heard, actually graduated from London University in the 1920s. I've been very lucky. My family have always believed in education, 100%. And that's really why I've managed to get to where I am today. I have to make some confessions here. Most of you are thinking, oh, he's a neuroscientist. He'll know an awful lot about the brain. Well, actually, my background is that I'm a microbiologist. I have a first-class honours in microbiology. I try not to tell the director of teaching that too strongly, otherwise he'll get me giving some lectures in that field as well. But I think a lot of that came from the approach. So I did my first degree at Edinburgh. And my first year I did biology, chemistry, physics and mathematics. I actually found out earlier this summer, so Jim Naismith and I did the same classes together. We didn't know each other or anything, but that was the approach. We did a whole range of the different disciplines. As a result of that, I've been very lucky to work with some really great scientists, not just because of the research they actually have done, but also because they really believed in mentoring people and helping people. And I always think that's a really important aspect that we should all do. In particular, as a summer student, I worked with this chap, Graham Reed. Some people in the School of Biology will know him because actually he tends to be our external assessor for our sort of teaching. He's now dean at sort of Edinburgh University for teaching aspects. And he was fantastic in that he sort of got me sort of like a summer job for 6 weeks. And it was very successful. I had my first publication as an undergraduate. As you'll see, I had a different name then. I was just Francis Gunn, okay? Some of the other people on here, you may know. Steve Chapman is now, of course, the principal of Heriot-Watt University. So even then, I was very lucky. I was working with some great scientists. As an undergraduate, I sort of I passed, and then off I went then to Cambridge. And this was the title of my PhD, the overexpression, topology, and purification of the L-fucose proton symporter, or FuCP, we used to say very carefully, of Escherichia coli. For those of you who are sort of a bit geeky, there is actually a kinase in this open reading frame as well, and that was always very embarrassing when we had to put that gene up. Anyway, it was a very interdisciplinary sort of PhD. These were the days when you did molecular biology and there were no kits. I feel really old, actually, when I say this now. Most of my crew will actually know this story. My first job as a PhD student was I was given a 2-kilogram tub of phenol and told, go away and distill it. with the only advice that if you get a 10% burn, you'll die. But we were expected to do that. I was expected, because of the background I had, to be able to understand the chemistry to go and distill it, purify it. So yes, there are lots of sort of like you purified your own chemicals and lots of maths. We used to do things called two-substrate kinetics, which even to these days, I found out we don't even teach that anymore because we feel it's a bit too far for the undergraduates. Anyway, the essence of what I did to sort of predict, using molecular biology techniques, which were very new at the time, the structure of this particular protein. And this protein is involved in transporting sugar molecules across the membrane of the E. coli. So my boss at the time was a chap called Peter Henderson, who was an interesting boss, actually. I'll leave it at that. But Chris Tate, who was the sort of postdoc who in the lab, he's now at the LMB, they were fantastic. They really sort of allowed you to explore your own ideas. And in our work, which we published in, I think it was 1992, we predicted the structure. And then I got an email from Peter in 2010, a paper which came out in Nature, which basically showed that they solved the structure of this group in Japan, and we were right. It only took 18 years. So that sort of approach, keep going when you sometimes feel that it's an insurmountable sort of target, is one of the watchwords with sort of science. Keep going. Postdoctoral is really when I started to get into the neurobiology world. I worked with this guy, Jeremy Taveray. Jeremy Taveray he taught me not only sort of like some great sort of biochemistry, but also cricket, seeing his brother used to open for England. And when I worked with him, we were essentially discovering these new factors that could keep nerve cells alive. I mean, this was a big deal, as you can imagine. I then sort of moved back to Edinburgh, where I worked with this rather stern-looking chap, okay, sort of Peter Brophy, and actually Stephen Tate. So Stephen, yes, I've been raiding your your Facebook page here, okay? So Stephen and I essentially worked with Peter here, and we discovered how myelin, this sticky substance that surrounds nerve cells, actually sticks to the nerve cells. And that essentially got my job here. Both of them allowed me to follow my own ideas, or they didn't ask. That's quite important. But everything I'm now going to tell you all came from one Friday afternoon experiment when I was actually working in Jeremy's lab and then sort of surreptitiously was working on when I was in Peter's lab. But it all came from one experiment. And that brings us really onto the focus of the talk, which is about dementia. This is the serious part, and I will try and put some humor in it. Dementia, if you're over the age of 50, is probably the scariest word out there at the moment. Dementia is a term that most people 15 years ago used to avoid. They would never say it. A bit like how people never used to use the C word, cancer. We have huge success now in cancer, mainly because it took a lot of money and a lot of effort to actually sort of try and actually start to understand these diseases. We're at this sort of stage now with dementia. I like some other sort of quips. My grandfather used to say this: growing old is not for wimps. Very true. And then I actually told this once at a sort of lecture I was giving, and somebody in the audience gave me a better one. You know when you're growing old, when every new sensation is a symptom. Big grim. But dementia is a problem for all of us, and it's a problem for the world. And it's actually a problem for everybody in this world, because either you're unlucky and you will have dementia in your old age, or somebody in your family will actually have dementia in their old age, or if you're very lucky and nobody in your family is affected, it's essentially going to affect your wallets. It's going to cost you a lot of money, because this is the money we're talking about now. It affects a lot of people. 30 million people at the moment have dementia. We get about 4.6 million new cases a year. In the UK, 850,000 people have dementia. This is going to double. There are some problems associated with actually how you sort of diagnose dementia, but those are actually improving a lot. In the sort of, let's say, North America, Europe, and Australia, things have actually become sort of pretty standardized in the way that we actually diagnose dementia. But obviously, the two sort of big countries of China and India, where you've got 2 billion people, they're actually catching up. They're actually admitting now they have dementia problems, and huge problems. This is not an issue that is just about the Western world, I hasten to add. This is a problem for the whole world, and in particular, the developing world. By 2040, we'll be up to about 80 million people with dementia, and look at the stretch of people who will actually be suffering from dementia from the developing world. Right? This is a problem that's not going to go away, and we have to deal with this. We have to deal with it for lots of reasons, not just the sort of social, but the economic. This costs the world $400 billion a year, 1% of GDP. It's the 18th biggest economy. That's the sort of levels we're looking at here. In the UK, it costs £23 billion a year to look after people with dementia. This is more than the cost of all these put together. I'm not saying we should take the money away, obviously, from those sources for stroke, heart disease, and cancer. 23 billion is a lot of money, or actually in Edinburgh, as we would say, probably 23 trams, or maybe a third of HS2. In the UK, a third of people over the age of 65 will die with some form of dementia in their brain. However, sometimes it doesn't manifest itself, and in itself, that's interesting, but we don't fully understand why. 40% of over 65-year-olds will experience some form of memory loss. If there's no underlying medical condition, this is referred to as age-associated memory impairment. This is normal aging. This is where you're going to get worried, right? Because this is normal aging, this is dementia. Okay, normal aging. Not being able to remember details of a conversational event that took a year ago. Well, that's, I think, probably everybody in this room. If you have dementia, it's recent events. Not being able to remember the name of acquaintance, their name is, that's normal. Family members. Forgetting things occasionally, frequently. Difficulty finding words, pauses and substitutions when finding words. A lot of people with dementia are very good actually at slipping in words to try and make up for the word they can't remember. You are worried about your memory? This is what most people say, I can't remember that my memory's going. That's not usually a sign of dementia. It's when your relatives and friends think you have a problem. There are different types of dementia. Dementia itself is an umbrella term. It encompasses a whole range of diseases. Alzheimer's disease is the most common here. Here, this is involved in the finding the right words, common forms of dementia, lapses of memory. This is the one most people have come across, but there are other versions. Vascular dementia. Here we have problems with blood flow to the brain. Could actually associate with stroke. Dementia with Lewy bodies, which is a sort of chemical change that occurs in the brain. They have problems with attention, with alertness and attention, rather than memory at first. In frontal temporal dementia, I think I need a better laser. I should have brought some. I've got quite a few lasers in my lab. Anyway, frontal temporal dementia, here personality and behavior are sort of initially more affected than memory. So there are different types, but Alzheimer's is the most common. Now, why is this the case? This is the case because different regions of your brain do different things. That's it really in a nutshell. Different regions of this brain are involved, let's say, in vision or balance or perception. The parietal lobe here, actually that'd be grand, thank you. So, the parietal lobe in particular is always interesting. We have Einstein's brain. It was lost for about 20 years. I don't say I've got it, but the scientific community have Einstein's brain. And it was lost for a long time, but his parietal lobe was much larger for some reason. We have no idea what that means. But when we're talking about dementia, it's the frontal lobe region, the temporal lobe and the frontal lobe, because this is essentially involved with consciousness, emotion, and memory. That's what makes you a human, is what I would say. The reason why we are absolutely different to every other animal on the planet is that this region of our brain is much more developed. You don't need to have a medical degree to sort of see what's actually going on. This is why I called the title of this talk The Shrinking Brain. This is a normal, healthy brain after somebody's died. You see, it's nice and full. It looks healthy. This is somebody who's actually died of Alzheimer's disease. What you're looking at here is massive loss of the nerve cells and all the support cells that are actually in the brain. The problem we've got here is this is when somebody died. What happened 30, 40 years ago? What's actually happening in my brain right now? That's a problem. And the issue we've got here is because your brain is encased with this, hard bone. How are you going to see through hard bone? The Alzheimer's disease it's called Alzheimer's because of this chap, Alois Alzheimer, who first saw this poor patient called Auguste, and it sort of described these symptoms, which we now refer to as Alzheimer's disease. The sad fact is that the prognosis, if that Auguste came in now to a clinic, is the same now as then. One of the reasons for that is basic pure money. We need more money. That's the bottom line. But what do we know? Post-mortem hallmarks of Alzheimer's disease. Alois Alzheimer, he didn't just sort of describe what was actually happening to Auguste. He actually looked at the brain. And he first described the features, the chemical features that we can see that are changing. And these are referred to as either what are called amyloid-containing plaques, and these are found outside of cells, okay? They form this like chemical mass, and you can physically see this mass in a diseased brain. We also have changes which are occurring inside the nerve cells, what are called neurofibrillary tangles, because a particular chemical called tau starts to go wrong. So that's occurring inside. But as I said before, the problem is we only know this having looked at somebody who's died. What happened 10, 20, 34 years beforehand? What we do know, however, because of various sort of genetic studies, is that the amyloid substance leads to Alzheimer's disease. So if you have a general increase in amyloid, then you can actually sort of contract Alzheimer's. So where are we at the moment? So how do you look for this? Diagnosing Alzheimer's disease is very tricky. What you have to do is rule out everything else and see what you're left with, essentially. The tools we have, most of you now would have heard of MRI. What you're actually doing in an MRI is you're actually looking at the volume of the brain. How much has it shrunk? The shrinking brain. How much is it going down? The important issue is never just to think of this in one snapshot. You need to take that image and then take another image 6 months later to see what the delta change is. There are PET scans. In PET scans, what you're looking at is glucose metabolism. Glucose is very important for your brain, and I'll tell you more about that a little bit later on. We now have modified PET scans, something called Pittsburgh compound B, which can actually, you can see the buildup of this amyloid substance directly in the brain. The problem we're having at the moment is that sometimes this increase as such like this doesn't always correlate, and we can't quite work out the correlation. Biochemical tests would be ideal. It would be lovely if you literally just sort of went to the doctors, took some blood, and they ran some sort of tests. There are no tests at the moment. Some are being sort of trialed at the moment. There are some sort of changes we think are occurring. One of the issues we're seeing, however, is that the biochemical changes we're seeing that change imply that a lot of damage has already happened. Doctors always want to diagnose something early. Well, you have a problem there because to be able to diagnose something, something has to have gone wrong. That's the problem. That's the catch-22. How early could you actually catch it to see that there is some damage to be able to get your test? Genes, of course, become much more sort of popular, certainly in the press recently. Early onset is a particular form of Alzheimer's, which affects about 1% of cases. These are genes which are associated in producing this amyloid substance. What happens is if you're unlucky to actually have one of these familial forms of Alzheimer's, then your brain starts to produce too much amyloid. Very rare. 99% of cases are what we call late onset or sporadic. There are genes now which are now being associated with this version. But people get very worried about this, and this is where mathematics comes in. Because these are the genes which are now associated. It doesn't matter what they all stand for, but these are the genes which, if you are very rare, you have a high risk of actually contracting Alzheimer's disease. And that's this early onset. What we're now getting in, literally I think it was 2 months ago, Julie in Cardiff has published a series of genes now, which they're very common, but the risk factor is very low. And as I always like to tell people, if you find out that at the end of the day you do have a mutation in one of these genes, if you walked a mile every day or did some exercise, you would offset that risk. So please, what I'm trying to beg you is don't get sort of paranoid just because of your genes, okay? Risk factors we do know of are age, obvious. Risk factor for sort of dementia, diet. Poor diet Poor exercise increases your risks for Alzheimer's disease. What are the treatments we have? These are the treatments that we have at the moment, which are essentially protecting what is left. So you will have heard of a lot of these drugs. What they're essentially doing is those nerve cells which are left, you're actually trying to crank them up as much as possible, get them working even harder. There are sort of drugs which are now being targeted for this amyloid substance, the processing of it, and also these neurofibrillary tangles I was telling you as well. The vaccines which are being trialled at the moment, which actually have all failed so far, unfortunately, this is essentially what they're doing. But then a new concept that my group is trying to sort of promote, and actually is now getting acceptance, is that you need to target particular vulnerable targets from toxicity. If you can understand the targets which are being affected and then protect them, then that would improve. We are never going to have just one drug. We are going to need drugs that do all of these combinations. So what are we doing? Here's the biology with a bit of chemistry. Okay, so amyloid, I've told you, is outside of these cells, and how does it kill cells? Well, it attaches to sort of targets on the nerve cell, which it elicits a sort of chemical reaction and kills the cell. That's what most people work on. I've actually done a little bit of this sort of work myself because, as some of you may know, or my friends will know, I like cats. All my cats have been named after particular malt whiskies, okay? No preference, okay, I shan't do that. But actually, I'm quite lucky. My wife is actually professor of feline medicine at Edinburgh University. There are only 3 in the world, okay? And we had a chance conversation one Saturday morning, and she was saying, well, our particular cat, Cardew, seemed to be showing some signs, neurological problems. And so that got us sort of like discussing, well, do cats get Alzheimer's disease? So Danielle basically had a series of cats which had come into a clinic, which had all been euthanized, which had all had neurological problems. But we didn't really know why they died. So basically, we started to do some staining. Remember these amyloid plaques? Well, if this is what a human sort of brain looks like with these amyloid plaques, this is what dogs, so dogs actually do this amyloid plaques work by Liz Head in California showing this. Cats don't really seem to do this. What they do is the tau. So here's a human, this is this neurofibrillary tangle aspect, the substance occurring inside the nerve cells. Human with Alzheimer's disease, dogs don't seem to do this, but cats do. So what that sort of told me is that we haven't really looked in other species enough. As human beings, we are very arrogant in thinking that we are completely and utterly unique. So actually, here's something for the future I'm planning to do. I've started the collaboration. We're going to look in other species. That is a dolphin. I was sent this sort of material, these slides, from some people working in Venezuela, in Maria. And these are dolphins from strandings. So why do we have strandings? So essentially what we're now doing is with help with the Rosalind Institute and actually Vincent Yannick at the SOI, we're now going to be able to get the brains from strandings of dolphins which have actually died, and we're going to actually see, do they get Alzheimer's disease? Other aspects we're doing is this substance amyloid, however, is, everybody says the dogma had always been it's outside of the nerve cells. But it turns out that amyloid is first made inside nerve cells, and we know it's toxic. So why would this be important? Well, if this is where it's first made, inside the cell, just as a thought experiment, that's obviously going to be the early stages of Alzheimer's disease. So how could that be? So we work on how does this internal amyloid kill cells. And we've identified in a sort of number of publications targets which this amyloid binds to inside nerve cells and kills them. One of them is called ABAD, stands for amyloid binding. We weren't very sort of imaginative in our naming. Alcohol dehydrogenase, something called cyclophilin D. The names actually don't really matter. I'm just going to really focus on this one called ABAD, because what does ABAD do? Well, ABAD normally makes energy for your brain. That's its purpose. In particular, under conditions of stress, ABAD is very good at making you energy. And it does this because it resides in what are structures in cells called mitochondria, sometimes referred to as the powerhouse of the cell. And essentially what they're doing is that they're making you energy. So when you eat sort of various food substances, glucose can be metabolized in a particular way. But other cellular substrates have to be broken down and used in different ways. So they make you energy. Now, essentially what we found was that amyloid can attach to ABAD, and we know that leads to cell death. But how? Well, I have to give you some, in the inaugurals, you have to give some embarrassing photos. So it seems all a bit like terrible cardigan. I have to give you a reason why this is important. Well, you see, as we age, things change. Actually, so that was me sort of like, I think last year, I tried to use the School of Psychology's sort of website. They've got an aging program because I thought I'd try and get a photograph of me even older. The problem is when I used it, I didn't look any different, which was a bit worrying actually. So actually I'm showing you a bottle of 1863 Madeira, which I actually have drunk. Anyway, so the important issue here is that when you're very young, your brain just uses glucose. That's what it uses. As you age, your brain starts to not use so much glucose, and it has to rely on these mitochondria to produce the energy. So as you get older, you have to use these mitochondria. You have to use ABAD. What we identified was that we'd worked out what ABAD looks like, what the three-dimensional structure actually looks like. And we worked out where the amyloid attaches to ABAD. Something called loop D. So everybody's got to remember loop D. When amyloid attaches to ABAD, it essentially switches off the activity, and it leads to the production of various toxins. What do these toxins do? Well, essentially what we found is that this is a mitochondrion, this is ABAD, amyloid's attached to it, and we've been finding out the consequences, what goes on. And what we found is that there are particular genes that get switched on because of amyloid attaching to ABAD. We still don't fully understand how they are connected, but these genes get switched on, not only in our sort of animal models, but in human beings as well. We've shown that. These genes, this one here, is an antioxidant protein. It's very good at protecting your brain. This one, however, seems to want to kill your brain. So you start to get this yin and yang. You get some genes being switched on which will try and kill your nerve cells. Some of them will try and actually keep them alive. You start to wonder, well, is that why if you take 100 people with Alzheimer's, they all look slightly different in the way they present? Because the genes that are all being switched on are slightly different in each case. So we've identified these genes which are being switched on, and we've identified that they're being switched on in human beings as well. Some of these have never been seen before, and so what they tell you is that you've got new chemical pathways being activated in Alzheimer's disease. These are places that you can start to think of making drugs. What are they doing? Well, in the case of one of these, EP1 or endothelin 1 here, endothelin 1 is really important in how nerve cells talk to each other. Nerve cells talk to each other by synapses. So if you have an interaction, one nerve cell to another, it talks to each other by this synapse. Electrical activity from one nerve cell is passed through the synapse to this one here. That is your memories. That is what a memory is. People ask me, what is a memory? That connection there is a memory. Now, the interesting thing is that in your brain, you have probably 100 billion nerve cells, and each nerve cell could have up to maybe 10,000 connections. You can start to see the complexity. But in essence, that's a memory. What happens in Alzheimer's disease? You start to lose your memory. Endothelin 1, we now know, is involved how these synapses are put together and how they work. So that's one aspect we're actually looking at. I've put this slide up here because other genes that we're looking at sometimes have come from rather different angles. You remember when I was working in sort of like Bristol with Jeremy Tavray, I was interested in how sort of like, when I was working with Peter Brophy in Edinburgh, I was interested in how sort of myelin attaches to nerve cells. Well, in the work I was doing there, I actually discovered a new gene. Probably the lasting legacy of my sort of career will be I got to name a gene. I named it Willin, all right, and Cell actually now call it. It's also referred to sometimes as FRMD6, but all of you in the audience will now call it Willin, right? I named it after this rather grumpy chap, okay, William Dick, who actually sort of set up the vet school in Edinburgh because that's where I was working at the time. Please, I named it after the William part and not his surname because that would have been way too rude. But we've actually been working on this, and one of the things we actually found is that this gene is actually involved in cancer. Published a series of papers in the last like 2 years where it activates what's called the hippo signaling pathway. We do love to give things silly names. But this is involved in cancer. But then actually a number of papers have just come out. And actually there are sort of 7 separate studies which have said that our willing is one of these new genes involved in Alzheimer's disease. So you can imagine Sylvia and I are writing the grant as we speak. So we've been discovering which genes get switched on and what they actually do, but we did something, I think, a little bit clever. You remember we sort of worked out how amyloid attaches to ABAD, and it attaches via this loop D. Well, what we did was that our poor old mouse models for Alzheimer's, we actually gave them this loop D. We injected them with this loop D, which binds amyloid, and lo and behold, these genes that were actually switched on got switched off. More exciting than that, we sort of published last year that these animals have learning and memory problems. Their memory got better. That tells you it's a drug target. That's actually how you find drug targets, by a very sort of quite a simple experiment. So we're now trying various methods to make drugs. So actually this, I think, actually there was, when I sort of published all this, you then get lots of press. How proud was I? Daily Express, Daily Mail. Actually, I called an end to this story when Richard and Judy wanted me to appear that evening, and I went, no, that's a step too far. But one aspect, so I mean, it was a real surprise because it's a little story, and it just went rocketed, and it was a bit of a shock because I literally went to the newspaper, and the guy at actually it was strange. I went down to the supermarket, and there was a homeless guy there, and he sort of said to me, "You're the guy in the newspaper." That's a bit strange when the homeless guy knows your name. So, chemistry. So we're trying to make drugs. Now, to make a drug is expensive. Making a drug costs 200, 300 million pounds, something like that. So how do you do that on a sort of small budget? Well, we're doing this by being smart. And one of the ways we're doing this is we're refining our drugs. So this loop D is too big at the moment. We're trying to make it smaller, because the smaller that you make a drug, the better, the more effective it could be. We're also developing a number of high-throughput screens as well, and I'm not going to go through all this technology, but essentially what we're doing here is using technology which was actually existed in St Andrews. I learnt about this technology because of the late Rupert Russell, who was actually in biology, and he was working on drugs actually for influenza. And so I heard him sort of like give a talk one time, and I went, that's smart. I wonder if we could actually adapt that to what we're doing. And we did. And we've actually got sort of drugs that we are trying to take even further. One of the things you actually do with drugs is that you have to work out how your drug interacts three-dimensionally with your target. To do that, we have to make crystals. Anybody did like alum crystals? These are protein crystals. We can do this because of the wonderful technology we have here in St Andrews. We have high-throughput technology for making crystals. That's all here. We are, as I say, making sort of compounds. Obviously, I can't show you what they are. I have to give you smiley faces, all right? But the beauty about all of this is, from my point of view, is you start to realize you're entering into a new world. You're entering into the world of chemistry. We're actually about to sort of pitch for one of the sort of drug screens at the BioCity, the new £100 million investment outside of Newhouse. And we're hopeful that they will actually accept this as one of the targets to screen for. We are using chemistry as well to look at the activity of ABAD in a completely new way. If you give this compound, doesn't matter what it looks like, okay, everybody's going a bit, some people here will be sweating a bit, saying he's talking a lot of chemistry now, but if you have ABAD around, it makes the cells go yellow. So here are cells, you won't be able to see them here, but if they're expressing our ABAD, the cells go yellow. Now what happens when you give them amyloid? They go less yellow. What happens when you give them your new wonder compound? Hopefully they go yellow again. That's a drug screen. Drug screen on a budget. So that's the realms of chemistry. What about physics? This is where we're getting into the science fiction world, right? So literally all of this came about from one email that Kishan Dalakia sent out one time. You see, light can do weird things. Some of you probably have seen this movie before. Light has momentum. This is the world's smallest golf swing. Right? These are actually about 10 micron spheres, and they're all being moved by light. Nobody's touching them. They're all being controlled by light. Actually, about the sort of light, the power in this is enough to actually move those, 10 microns, beads. So if you think of your sort of Star Trek, and you remember your sort of tractor beams, that's actually true, but on a very small scale. So light has momentum. So if light has momentum, it means you can do things with it. So what would you want to do with this? Now, you remember when I was working in Bristol with Jeremy Tavray, and we were working on the neurotrophic factors, what essentially we were doing is we were taking nerve cells and adding a neurotrophic factor to them, and then getting them to grow in particular ways, and identifying all of the chemical pathways which were being activated in this to see if we could actually modify them, actually regrow nerve cells. And then we started to do some really fanciful experiments, because what we did was we used lasers to do this. You're looking at a growth cone here, right? And that's a laser spot, and we're getting it to grow towards the laser spot. You can manipulate nerve growth by just using light. Most remarkable thing. For those who are for the science side of things, this is not heating. We know that. We measured it. What we're now realizing is that cells don't just respond to lots of chemical cues. They respond to physical cues as well. The stem cell world have actually woken up to this. There are actually the sort of physical environment of a cell is just as important as the chemical world. But how about that? So what we're actually now doing, I've actually sort of now got literally arrived about a month ago, new DNA constructs that will be able to allow us to actually give us much more control over this. Other things we did, and some of you may have seen this before, in that we used laser light in a very specialized way to what we call the optical syringe. So what we were able to do was that we're able to use laser light to punch tiny little holes in cells. So what we can do is that we can actually sort of punch holes into cells, and then in this case, we put in these actually primary neurons for any neuroscientists here. This actually, we just published this 2 weeks ago, all right? We can actually put genes into primary nerve cells, or you could have put drugs in. When we first started publishing technology, our good friend the Daily Mail likened us to sort of Ewan McGregor. Okay, that's good. We went further in that we actually developed this technology so that your mobile phone can do it. What you're actually doing here is this is a touch screen. Those are cells. Where you touch those cells, right, we'll put a little hole in that cell, which it recovers from, and put your gene or chemical of interest in there. So the beauty of that is that I could do this here, and it's controlling the technology, which is literally just across there. Purely, the beauty of this is that you don't need to understand any of the mathematics, any of the physics which is behind here, but it actually, from an end user point of view, allows you to give you control that you could only have dreamt of. Now, we've done this sort of technology so that even people who would like to become the new Prime Minister of Scotland could actually use this as well. But that's the whole point of it. When we're developing a lot of this technology, we can actually use it so you don't need to worry. Now, in the case of Alzheimer's disease, this will give us a new way of potentially trying to treat cells, or testing our drugs. We've gone further. We've now put the ability, we can put all of this on the end of an optic fiber, the width of one of your hairs. I kid you not, if you've ever seen a neurosurgeon do neurosurgery, you would never want to have your brain worked on again, because they actually use quite large, actually to about this sort of size, pen size, sort of going into your brain. We've actually put this on the end of an optic fiber. So now that you can see what we're thinking of doing, I want to go into the brain. If you had asked me, could we ever use this technology and treat in a living brain 5 years ago, I would have said, no way, no way. I think we might be able to do it. And the reason why we can do that is that we've been now developing the optical fibers, which you can see 100 microns, and with specialized types of actually lenses on them, so that we can now image and treat all on the same optic fiber. That really has become sort of like science fiction, but I do think it's possible. We're already starting to try and do this, so we have people actually, we can grow slices of brain, which sounds a bit Frankenstein, okay? But we're seeing if we can actually sort of treat these slices of brain using these optic fibers. You can also use this sort of technology in a completely different way. You remember I sort of said one of the problems we have is we have this rapidly increasing number of people with dementia. Why? Why is that the case? The thing is because we've been hugely successful, and that is a good thing. People live longer. That is a very good thing. But the result of that is that we have a huge increase in the number of people. So by the time I sort of retire, we're going to be sort of over the 8 billion mark. The problem we have, by 2030, we won't have enough land to feed that people. That's fact. There will not be enough land to feed the people on the planet. You could argue at the moment that doesn't occur at the moment anyway, because of the way everything's distributed. But that is a problem. So we need new ways of actually sort of coming up with being able to do this. And one of the ways of doing that is, of course, things like genetic modification. Can you genetically modify crops? And in particular, we're talking here about the monocots, these cereals, the barley, the rice, the wheat, because this is what feeds the people. They're very difficult plants to actually put genes into. They're very difficult methods. So we published this about a month ago. So this is what we're now doing. These are plant cells, where we're treating them with laser light to be able to put chemicals into those plants. As this, we think this could be a brand new way to actually modify plants, as a way to actually help feed these people and therefore help dementia as well. It's interesting when I sort of look back how everything interconnects to some extent. Will we ever cure Alzheimer's disease? Because that's really what you want to know. I think we are on a cusp at the moment. Governments are waking up. We have a G8 summit on dementia happening in the next week. This is fantastic. This wouldn't have happened 5 years ago. Governments are starting to wake up that this is a problem that is not going to go away. This is a problem that is going to cost us a lot of money, and we have to do something, and we have to do something now. I really do believe that we are making huge advances. We really are. Things are being tried at the moment. Some of them will fail. That is the nature of the beast, I'm afraid. It will take time. The reason why we actually sort of done so well in cancer was because of the sheer money that was actually put into this. At the moment, in the UK, we spend £11 on research for people with dementia. It's not a lot, is it, really, when you think about it? But there are advances. There are new vaccines which are being trialled at the moment. Who knows where they're going to take us? There are new compounds being tried at the moment. I heard Giovanni Malucci give a talk from Leicester. She's actually been working on prion diseases, and she was talking the other day about sort of this new sort of drug she's got, which seems to be very good at protecting nerve cells from dying. She knows at the moment that if you took that drug, it would actually probably give you diabetes. But it tells you it's a starting point. It tells you that it might be possible. What we have to do is then start to modify things. You can never predict where things are going to go. And that's really my answer when people ask Alzheimer's disease. If you had asked me 10 years ago that you could take a gene that exists in Chlamydomonas, lives in pond life, right? You probably all looked at it when you were at school under the microscope. Swims around, very pretty. These very small sort of organisms have genes in them which actually make particular molecules that sit in your membrane, that sit in their membrane, that when you actually shine particular wavelengths of light at them, they open up so that ions can flow. So you can actually cause an electrical charge to be produced. So we published this last 2 weeks ago, where what we've done is that using our photoporation technique, this laser light, we actually sort of put one of these genes into primary neurons, and now you actually give them particular light, and you get electrical activity. So what that tells you is that you can now use light to activate nerves. So let's be really fanciful. If I told you that it might be possible to actually use light to create a memory. Would you believe me? Probably not. It's been done this year. It is now possible to actually give an animal a false memory by just using light. So who knows where we may be able to go in the future? Because it's really quite remarkable where we've come from. Another example, it took them 13 years to do the first human genome. They can now do your genome in a day. Technology is going at a tremendous pace. And I really do think we will have drugs in the future that can help. So to conclude the sort of message I'm giving you here, this is all about interdisciplinary research. This is the way to tackle things for the future. This is the only way. We use biology to understand the biochemical changes. You've seen we use chemistry to make new drugs. We're using physics to develop new technology. I try and tell all my guys this. This is really important. Never be afraid of the approach. Some of my old bosses used to say to me, oh, no, we don't do that. That's not our approach. And I used to say, why? You can always find out somebody who actually would do it. That's, for me, why I've loved being at St Andrews, because of the wealth of expertise we have in all of the disciplines that we have. Answer the question, and imagination is critical. I always tell our sort of students, there's no such thing as a stupid question, only a stupid answer. It's important just to have the idea first and then work out how you could do it. I have many people to thank. Yeah, so it's terrible photo, okay. Obviously my family, in particular my mother, I should say, because I am actually wearing the suit my mother bought me when I graduated as an undergraduate. Are you impressed or what? What I should say is you buy a good suit which can be let out. Right? Obviously my friends, this is them celebrating the best champagne tasting we ever had, wasn't it? It was something else. My research colleagues in biology, chemistry, physics, medicine, psychology, and neuroscience. A particular mention to Kishan, who's in Taiwan. He's always traveling. But Kishan and Wilson and Ifer and Thomas, who really opened up a new area for me. You know, when I first sort of thought, I got this strange email, we can do some strange things with lasers. Is anybody interested? And I was a bit, because it was going to be a risk. It was going to take time. But boy, look at what it's actually produced. The general public and their funders, all you lot, you are the people, you pay your taxes, you donate your money for people like myself. I do say I have the best job in the world because I'm paid to have ideas. How privileged is that? It really is. Number of people as well I would like to thank, obviously, all the sort of secretaries, and in particular Sandra Murray, if she's around here, because she's looked after my grants for many years and made sure that we haven't overspent and make sure that we keep everything on the straight and narrow. Most of you will probably know I live in Edinburgh, so I have done a lot of miles. I've seen a lot of sunrises and sunsets with all of these people here, and we have a saying, whatever's said in the car stays in the car. Good therapy, right? I was sort of saying actually before that the car has actually probably qualified 6 PhD students of its own, not my own students, but just people I've given lift to. You probably noticed at the beginning, my first paper was actually Francis Gunn. Best collaboration ever made, my Danielle. But I didn't get where I am today without this lot. The reason that professors get their chairs is because of the people who do all the bloody hard work. One of my guys told me a month ago, they stayed up overnight to actually purify probably the largest amount of ABAD we've ever produced. And they did that because they love the work. This is my current group. They are an interesting bunch. I should show. It's all right, don't worry, it's not a virus, don't worry. Over the years, I've been very lucky because I've worked with a whole range of different sort of people. And what's been fantastic about them is that they've all sort of like, they all turn up, they're always cheerful, which is nice. But what I love about it is that when I look at them, you have biologists, chemists, and physicists all working on the same project. So with that, thank you very much. And I'm going to leave you with one quote which I always like. There are 3 stages in a scientific discovery. First, people deny it's true, then they deny it's important, and then obviously they give the credit to the wrong person. And with that, thank you very much.